Welcome, everyone. Good morning or good evening, based on where you are. So today we'll be going through first of our series using JupyterLab on UK Biobank Research Analysis Platform. So my name is Anastasia Sedlakova. I'm Senior Community Engagement Scientist at DNA Nexus, focused on UK Biobank. And today I will have my colleague, who will be my Watson to make it a bit more interactive. So Ted, if you can introduce yourself.
Hi, my name is Ted Ladaris. So I'm a principal scientist and I'm a bioinformatics trainer at DNA Nexus.
So you will meet with Ted more next week when you will see our second session. So let us, before we will start, just said to you that it's a webinar series. We already have overview. You can click on the link and see the record of overview session. Today we'll have introduction into Jupyter Notebook. And next week with that, you will be doing more in-depth analysis or looking into Jupyter Notebooks with UK Biobank data. Okay, so what are our learning objectives for today? So we should be able to explain the relationship between JupyterLab and the UKB-RAP project, especially regarding storage. Select the appropriate JupyterLab instance given your workflow requirements. Access files and Jupyter Notebooks from a project into JupyterLab environment. Run a Jupyter Notebook in the JupyterLab environment and upload results back into the project, and utilize and install software dependencies into JupyterLab environment. So here is a TLDR section, because we all have not much of time. So for you, if you have to remember, like the most important thing from this webinar is that UKB-RAP has two types of instances. So single node JupyterLab, you should use it when you have already extracted data into a file. So you don't need to use distributed computing, or you want to use data. Then we have a Spark cluster JupyterLab. You should use it when you want to query dataset directly, perhaps using some complex queries, or you need to use Spark-based tool for genomics, for example, like Halo and Glow. And the second very important thing, actually maybe more important than the first, is that UKB-RAP has two types of storages. One is research analysis project storage, and another is JupyterLab storage. And specific thing on JupyterLab storage is that everything in it will disappear once session will end. And we'll talk a lot about it in the next few moments. So we'll have a lot of information, and then we'll have a quick demo in the end. So just wait for the demo. Okay, so here is the slide that you will possibly remember from overview session, if you were there, that there are different ways how you can interact with UKB-RAP. So you can start by using UI, when you can create project, you can assign different roles for your collaborators, you can add files, you can look at the files and organize them. Then you can also use Cohort browser, and we actually will be using Cohort browser today to create cohorts. Then you can use apps, for example, to analyze using Swiss Army knife. And then there are two different types of JupyterLab. This JupyterLab, we call it single node JupyterLab, and here is Spark JupyterLab, that you can interact with both of them. So this lecture will be focused on this first JupyterLab, and the section next week will be focused on Spark JupyterLab. So actually, I was repeating JupyterLab word quite a lot, and we saw that a third of you never use it. So what it is a web-based user interface where you can work with Jupyter Notebooks, and Jupyter Notebooks are actually documents then where you can have like you can have code, you can have text, equations, images, and interactive visualizations. So this is the type of tool that you will be using if you use Python and if you are doing data analysis in it. So here is just a print screen of JupyterLab and how it looks like. So here it is, the JupyterLab, it's the Jupyter Notebook itself. Here you can see a cell and the output of the cell. Here is a toolbar, and here is a left sidebar where we'll see different files. And sorry, I catch a cold, so it's very good that we're all online. So, because we saw that we have different, like 2 types of JupyterLab instance, how you can choose between them. So this depends on what kind of workflow are you using. So you should use single node JupyterLab instance if you do not need access to Spark, data are extracted into the file, for example, TSV file or CSV file, and you can usually run some Python or R code in it, and you can do some more heavy machine learning or image processing, or you can run code in Stata. Use Spark cluster JupyterLab if you want to leverage power of Spark for distributed computing. You want to query the dispense database directly. For example, you want to create more complex queries than you could do in a Cohort browser, or you need to use Spark-based tools such as DXData for querying. We will look at it, or you will look at it next week, Hail and Glow.
Quick question, Anastasia.
Yeah.
So what point should I switch over to Spark if I'm used to using just kind of single JupyterLab?
Mm-hmm. Yeah, so in case you're used to use, actually, we will be showing workflow where you don't need to use Spark at all. If you are just using phenotypic data, let's say that you're an epidemiologist and you want to conduct this research, you don't need, for example, whole exome data, you can just use single node JupyterLab and never use Spark JupyterLab. But if you want to make some complex query or do some more advanced bioinformatics or use genetic data, then you will definitely need to use Spark JupyterLab.
Great, thank you.
So when we will be talking about different JupyterLab instances, we will be seeing named kernel and feature, and actually what's the difference between 2 of them. So there is the term kernel, which is used in JupyterLab, and it's a program that runs and introspects the user's code. So you need to select kernel based on what programming language you want to use. For example, if I'm going to write code in Python, then I will select Python kernel. And what are the available kernels in UKB-RAP? So Python, R, and Stata, but Stata only for single node JupyterLab. And then we have another term, which is feature. And this is a term used in UKB-RAP, which is a set of kernels with pre-installed packages, such as pandas for data analysis, or NumPy or matplotlib for plotting. And available features are for single node is Python or R, then it's machine learning or image processing in Stata, and for Spark cluster is Hail, Glow, or Hail and VEP.
Quick question. So I'm already confused. So why do we use both of these terms?
Yeah, okay. Yeah, so this is confusing. So the thing is that in a few moments we will be launching JupyterLab, and to launch JupyterLab on UKB-RAP, you need to know what feature you want to use. And then once you launch your JupyterLab and you want to run your Jupyter Notebooks, you need to know what kernel to use. So that's why we're using the 2 different terms. And actually here you have the hierarchy. So for example, here we're choosing feature Python and R, which is actually what we will be choosing for today's demo. And this feature contains 2 different kernels. So either I can write my code in R or I can write my code in Python.
Great, thank you.
Okay. So we were talking about the difference or different use case for JupyterLab. And here are different use cases, but just like more described. So we have a single JupyterLab for Python and R. So the feature, as I was telling a few moments before. Then we have another use case as a single JupyterLab for Stata, or a single JupyterLab for machine learning and image processing. And here you can see that the different libraries pre-installed. Here are the use case for Spark JupyterLab, when you can see that it's either, for example, use case for customized cohort querying data using DXData or PySpark or Spark SQL. Or if you want to do some more advanced scalable genomics, you can use Hail or Glow. And you see that all of the use cases you can use with non-interactive execution with Papermill. And Papermill is a very interesting tool how you can automate your Jupyter notebooks and run them non-interactively. So the use case would be that you run some analysis on one data file, you clean it or you convert it to another format, and then you said, okay, that works. So you want to write it scale, you want to run this code on the different files. So usually what you will do is you will need to rewrite your code from Jupyter Notebook into just plain Python. But with Papermill, you don't have to. So you just need to parameterize Notebook by adding text to cells, and then you just automate it like that. Okay, so before when Ted was asking, I was mentioning that there are two different types of workflows, how you can use JupyterLabs. And one, let's say, is avoiding Spark, and another is embracing Spark. So this one is when you are not using Spark, when you are using Cohort browser. And this is actually the workflow that we will be following today. So we'll use Cohort browser to build cohorts. We'll then export cohort fields into tab-separated values using Table Exporter app. Then we will load this file and analyze it in JupyterLab, and then we will export results to project. And here it's the one, advanced querying or embracing Spark. So you create cohort using DXData, PySpark, or Spark SQL. Then you retrieve cohort fields and save data frame as a file. Then you load and analyze data in JupyterLab and then export results to project. So this part is done in Spark JupyterLab, and this part is done in single node JupyterLab. But it could easily be done everything together in Spark JupyterLab. It's just because of the cost. So if the analysis that you are doing is very long, then based on like cost consideration, we recommend you to do filtering and cohort creating in Spark, and then use just single node JupyterLab to save money. But you can also use Spark for all if you want to use Python or R. So things to do before opening single node JupyterLab. As I said, prerequisite for a single node JupyterLab is that you have to have your data extracted in the file format. And since I was showing a few slides before, there are two different ways how you can do that. One is that you can do it using DXData or PySpark or Spark SQL, and Ted will be covering it in depth next week. So if you're not registered on this webinar, I encourage you to register. Or you can create cohort and using Cohort browser, which we'll be doing today, and then export cohort into file using Table Exporter. And both of these different like set of steps will end up in some data extracted in file format that we can use in our single node JupyterLab. Okay, so let's start our workflow by starting Cohort browser. If you are new to UKB-RAP, you will not find it as a tab or something. So once your data were dispensed in your root project, in your root directory, you will see something like that, dataset. You will not see these things because you need to create cohorts. So you will see just dataset in a freshly dispensed project. You click on the dataset and the Cohort browser will be open in next tab. So this is how the Cohort browser look like. And here are the different components of it. So here is a cohort created using filters. Here we'll be doing some data exploration, and here you can do comparison. So how can we explore data? We can explore data by clicking on Add Tile button, and then you will see something that we called Field Explorer. You can explore fields that are available to you based on the application that you submitted. So for example, here we're searching for genetic sex. So we're typing the name of variable here, and you will see that there are different. So it's case insensitive, and there will be different variables, and you can see some additional information, for example, link to UK Biobank, some basic statistics, what is the value type, and what entity it is. So let's create the cohort using filters. So how can we do that? We can start by clicking on Add Filter. Then it takes us to Field Explorer that I showed you before. We're searching for sex variable because we want to create female cohort, so only female will be there. We then click on Add as Filter, and then we're adding condition. Is any of female, will create a female cohort. So you see that out of 500K, it's roughly 1/2 is a female cohort. So how can we add fields to cohort? We then need to go to Data Preview section, and then we're actually will be clicking on this table icon, let's call it like that. Then there will add columns, and then it again takes you to Field Explorer. When we type, for example, months of birth, and then you see that the different variables appear. We're interested in months of birth. So we click Add as a column, and we're having our EID, which is automatically dispensed, and month of birth. So once you created your cohort, don't forget to save it. Then you can then use it in Table Exporter. So we created cohort. Let's move to the next step, Table Exporter. How can you find Table Exporter? You go to Tools section, Tools Library, and then you search for Table Exporter, or you scroll down. And then what you need to select is Dataset or Cohort or Dashboard. So you click on Select Record, and you select the cohort. So we'll be selecting here female cohort that we just created. Here you will see Add, and so you see a female cohort here. So we can then specify what we want to see. If we want to see what format will be, CSV, what will be, if it will be data.csv. So you can be more detailed by specifying what your file name. Then there could be coding options, if there will be coding, or there will be raw data, and header style. So field name will be something like P31 for sex, and field title will be sex, so the human readable name instead of a code.
Quick question, Anastasia.
Yeah.
So why can't I just extract all of the fields from UK Biobank? Why do I have to select fields?
Yeah, so this is a good question. It could be like very exhausting to select field by field. So the thing is that we have, for phenotypic data, we have, if I'm correct, more than 40,000 of different fields. And if you will export this into table, it not only will cost a lot, but the table will be huge. So you need to select carefully your data in order to be it manageable, because by clicking like that, this will be the job that will cost hundreds or thousands of dollars for it. So I suggest you to explore your data set, to select what you want to explore, and then extract it into file.
Great, thank you.
So before we will be launching our Jupyter Lab, I just want to emphasize the differences between running local analysis versus running analysis on cloud. So when you run your local analysis, there is your computer with some software environment that you installed. Then there is computer that you own, and then you control, hopefully. So you have some analysis code that you need to bring into your computer, or you have it already there. And also there are some data that are either on your computer or you need to load them there somehow. You make some computation and you produce results, and everything is in your computer. Whereas in cloud-based analysis, by requesting machine, you are requesting for some temporary computer somewhere in the cloud. And because you're requesting it on UKB-RAP, we have it packed with some pre-installed environment, for example, Python, R, or Stata, and with some libraries. So once your temporary machine is ready, you need to transfer data from project and some analysis from project. You execute code or your produced results inside your temporary computer. And then once you're satisfied with your results and you want to use them or to save them, you need to transfer results back to project. And this will be our key thing that we'll be returning, that it is your temporary computer. And once your session will end, this computer will disappear and all of the data there will disappear if you will not save it into your project. Okay, so now we know the basic differences between local and cloud analysis, and we can look more into how you can launch JupyterLab. So you click on the new JupyterLab. So you go from Tools, JupyterLab, and here you will see your form that you need to fill. So yes, a new JupyterLab. So there is environment name. So this is the name of your job. You can be specific or you can write test all the time, depends on if you want to reuse this configuration later. Then you need to select project. Then you need to select instance. So what type of temporary computer will you have? And also duration. So here we have a default value for 4 hours, but don't be afraid. If it will be like, if you want to terminate it early, you can do that. Or if you want to extend it, you can also do that. Then there is a priority. So we recommend to use high priority for JupyterLab. And then there is a configuration, either single node, which is the focus of our today's session, or Spark cluster, which will be covered by Ted. And then feature. So remember set of kernels. So here I'm showing Stata feature. Once you select all of these, the pricing will be estimated based on what you selected, and you can then start environment. So there is a note of those of you who will be using Stata feature. So you must have a valid Stata license, and you need to upload it into your project directory in JSON format. And then you should choose Stata kernel when launching JupyterLab. So once you will get this presentation slide, you can look into documentation, and there is a tutorial how you can create a JSON with your license. So we launch our JupyterLab, but it will take some time to start the instance. So how can we monitor it? So here you can see a status initializing, or you can look it in the job in monitor. And it will require some time, so you can meditate, make a stretch, drink coffee, make some short yoga stretch. I mean, like not 90 minutes full yoga class, but just 5 minutes yoga stretch. And then your JupyterLab will go from initializing to launching to ready. And once it's ready, you can click on single, for example, here the name of the job, and it will be opened in the next step with JupyterLab. If you will click on it before, it will show you some error because the computer, your temporary computer that you're renting, was not prepared at that time.
Hey, quick question. So what if I accidentally started like the wrong configuration? Like I was really enthusiastic and I accidentally started a Spark configuration. How do I fix that?
Yeah. So this is a good question. So what you can do is that, so when in your JupyterLab session or window, you can also end an environment. So if you click on end environment, it will be end. It will your Spark JupyterLab that you accidentally turned on or launch it will be ended, and you can then click on JupyterLab and make the configuration that you want to actually run.
Great, thank you.
Okay, so now working with JupyterLab, so we're going closer and closer to actually work inside of JupyterLab, and we're actually closer and closer to the demo part. So for us, in order to understand and to leverage and to use properly JupyterLab on RAP, we need to understand that there are two different types of storage, and I will be repeating it multiple times, and Ted will be repeating in the next session multiple times, that we have two types of project storage, two types of storage. One is research analysis platform project storage, and another is JupyterLab environment storage. And all of your analysis that are stored here. So we want to transfer them. For the Jupyter Notebook, it's done automatically, but for other files, you need to use DX download and DX fuse, and I will be looking more into details what it is. And then once you generated some files inside your JupyterLab environment, and you want to keep them, actually, you need to transfer them back to project, or transfer them to project using DX upload command line utility. And we will look more into details how you can actually do that. So the notebook that I will be showing here is a part of Open Bio repository, where we're putting example Jupyter notebook. So you can look there, and you can work with this Jupyter notebook. This could be a nice start when you're just starting in UKB-RAP. So as we were talking before about different two types of storages. So here is a JupyterLab storage. You will see it on the left sidebar. There will be a folder icon, and then there is a project storage, permanent-ish storage, because I said permanent-ish because you can also delete files there, but the files will not be deleted themselves. And the tab is called DNA Nexus. So this is the main part of this presentation that you need to remember, is that everything in JupyterLab storage will disappear once the session will end. Everything.
Great. So question. So I've used JupyterLab on other systems, and it just saves, and I've created notebooks and I've saved them, and it stays around. So are you saying after I terminate this JupyterLab instance, it's going to disappear?
Yes, this is what I'm saying, that we saw that some of you use JupyterLab routinely. So in case you are used to create JupyterLab, what could be this is not a rocket science, I just create it as I usually do this, and you will not upload it to your RAP project storage, it will really disappear. So here we will be showing you in the next slides how you can create the correct version of Jupyter Notebook so it will not disappear.
Great, thank you.
So this is the way how you can create a new notebook in a project storage. We will actually not show you how you can create notebook in a usual way, because we want to emphasize that this is the right way how to create Jupyter Notebooks on UKB-RAP. So here you will see a DNA nexus name in the tab, which is not part of usual JupyterLab. And there will be a new notebook. So you click on the new notebook, and then you need to specify kernel. So this is the way how you can create notebooks. As I say here, it's safer overall, but don't forget to save. So multiple times, click on the save icon to save your code and your results. So how you can open notebook from the project storage? As I was saying before, when we have two types of storages, and UKB-RAP for the Jupyter Notebook, you don't have to actually transfer your code from project to JupyterLab, because it's automatically done by just going to DNA Nexus section and just clicking on the Jupyter Notebook that you would like to open. Now, the one thing that I want to note is that when you save, when you click the Save button in JupyterLab, the notebook gets uploaded to a platform as a new file. So you will, in case that, for example, you share it with your collaborator, this new notebook will be, it will be written that it was created by you and not your collaborator, because it actually overwrites your previous Jupyter notebook. And if you want to see old versions, they're going into .notebook_archive, and this is the folder that you will see in your root directory of your project. So what are the components of a notebook, of Jupyter Notebook? So this is the notebook itself. So what is important is the active kernel. So here you see that we're using Python 3. Then there are also some icons that we can use, is, for example, save icon, add cell, or execute code cell, or you can use shortcut Shift Enter. Then there are different cells or code blocks. So here is a code block, and here is the output block. So here we're looking into data frame, and we want to make some summary statistics. So here the output is summary statistics. So the also very important thing is if, for example, as here, it's only less than 30 minutes left, and I want to update duration. I'm thinking like, oh, I want to have more time for my analysis. Then you can click on Update Duration button, and then you can set how many hours you want your notebook to run. So it will not add, so it will not be 2.5. It will just overwrite it to 4 hours. So let me show you basic notebook analysis workflow that we will be looking at the summary, then we'll look into details, and then I will show it to you in the demo section. So first, you can access a data file and load it into Python. And there are two different options how you can do that. Then you can perform analysis, get tables and graphs. So since this is a toy example, so here we're just doing basic filtering and making summary statistics. And once you want to actually save and keep your results, you save them as a file and you upload them back to project. So here is an example workflow what you can do on a single machine or single node JupyterLab. So you open or you create notebook, you download or access data to JupyterLab storage, you analyze them, you upload results to project, and you save notebook. So let us look more into details about the different steps that I already described. So first is accessing data, and there are two different ways how you can access them. So first is using the DXFuse file system and load it into pandas as usually using a read CSV function. So the DXFuse file system is actually adding, like you can use it by adding /minute/project to the path to your file. So by adding that, you're actually using this file system. The thing is that it's currently in beta version. It's for read-only, and it do not reflect recent changes in file system, meaning that if you launch your Jupyter Notebook, you created some files, you uploaded it to your project storage, and you wanted access during the same session using the DXFuse file system, it will not be possible. So this is just the things to keep in mind. The other way how you can access files that are in a project storage is that you can download files to JupyterLab environment. And you can do that using command line utility dx download. But wait, we were in a Python. So we need somehow to tell to Python that we want to use bash there or command line. And we can do that by using %bash magic command. And by writing this, your Python kernel understands that now you want to use command line, so you can write dx download /data/result CSV, and this will be downloaded into your local or like in your JupyterLab storage, temporary storage.
Quick question. So I see you have two different methods of getting data into JupyterLabs. So which one should I use?
Yeah, so generally we would recommend to use the first one, the DXFuse file system, but it could be slow if you have a big, like large data sets. So I suggest you to start with DXFuse, and then if it will be slow, then just use DX download.
Great, thank you.
So then we can do analysis, import data and do analysis. So here we're showing you the second option that you can do is, so we downloaded our data set into JupyterLab storage. So remember, the folder icon is JupyterLab temporary storage. We used DX download, and by this, actually, we see now that, that we have results CSV in our on our JupyterLab storage, and we can then read it normally as we would do with any locally stored file using, for example, pandas read CSV. And the last part of our workflow is upload notebook or data back to project. So we highly recommend that you will never use upload notebook option actually, because you already created notebook the correct way, and it's already is in the project storage. So you're uploading your created files using DX upload, and in the same way as we were using DX download, we use magic function %bash, and then we're writing DX upload your file name and pass to the data to the folder where you want your file to be uploaded. If you're collaborating with multiple people on a notebook, you can experience the situation that you will see this lock sign or your collaborator, and you cannot, if for example, Ted and I are collaborating, and Ted start to do some analysis, he want to make our notebook even better. Then when I want to access it, I will see this sign. And if I want to edit actually this notebook, then Ted need to actually shut down kernel, because only one person at a time can collaborate. So once Ted is done with all of the changes he wants to do, he click on the right click on the left bar menu, then he shut down kernel, and then I will see this notebook as available. And the last part that I want to cover before we will be going to the demo part is that there are some suggested best practices for installing software. Is that it's better to install using URL or using packages with package manager. So for Python it will be pip, for R it will be conda, and use version tags when possible. And if you want to utilize the libraries that you already created, you can use image snapshots. By this, you are saving software dependencies in reusable format, which means that once you install some packages, you can go to DNA Nexus section here on the tab, and you click on Create Snapshot, and the snapshot is saved in the root project directory in .notebook_snapshots. So next time you are launching your JupyterLab, we were not covering this part, but you can here select your snapshot, and your libraries will be already installed when your JupyterLab will be started. Okay, so let me go to the demo part. So I will log in into my UKB-RAP. And here, if I want to, so usually you start with the list of the projects. So usually you will have just one project, and then you click on a tool, and there will be JupyterLab. So then you click on New JupyterLab. I will make it. I will not start a environment because it takes like 5 to 10 minutes, and I had already started, but I will just walk you through what you can select here and how. So let's go to test. Then let's click on the project, select project. And then if I would have a snapshot, then I can select it. Then I will select instance type. So once you open here, you will see that there are different configurations. So how many total memory it has, how many cores, and how many total storage. Then there is duration, and there is a priority. I really do not recommend you to experiment with priority for JupyterLab, because if you are not requesting high priority, then it will be cheaper, but it could be that the notebook will disappear because of the differences in settings. So definitely choose high priority. Then there is cluster configuration. So here we'll be using, for today's session, single node, or you can use Spark cluster, but here we'll be using single node. And here I can select different features. So Python, ML, or Stata. I'm using Python. So based on that, the price is estimated, and then I can start environment. But as I said, I will not start because I have already here. So I can use, I can click on my JupyterLab name. So here it's single node JupyterLab, and JupyterLab will be opened in the next tab. So let me just open it. And for those of you who are routinely using JupyterLab, it looks almost like normal JupyterLab, except for this part and this part. So, as I said, if you want to create new notebook, please use DNA Nexus new notebook. We can do that. So by creating a new notebook, and here in our DNA Nexus storage, in our root storage, there is the notebook that we created. We can then rename it as test. And then when we can open it, but once we will be opening it, we'll be prompted to select kernel. So either it will be R, Bash, or Python. Let's leave it as Python. Okay. So I wanted to show you the workflow example that I was showing in my slides, and it's in OpenBio repository. So how you can do that? For these purposes, we just use normal launcher. So new terminal, and the terminal window will be open here. And we can use git clone. So I will not run it actually because I'm already downloaded repository here. And here is our, just to emphasize. So folder icon is temporary JupyterLab storage. DNA Nexus part is RAP project storage. So by doing this thing, I'll just copied all of the repository content into my JupyterLab storage. So I can open my JupyterLab example, and I can, for example, what I can do is I can load pandas because we'll be using pandas for data analysis, and I can then access data using the XFuse file system. Or I can download data, and the data will be using DX download, and the data will be here in the JupyterLab temporary storage. So either ways are okay, as we were talking. Then I can do some description, some descriptive statistics. Then I want to filter actually that I want to figure out what is the 95th percentile on the year of birth of my participants, and it's 1966. So I want to select younger participants. So I want their year of birth to be later than 1966. And so here you see that it will be at least 1967 and later. So here you can see that apart from that, out from 500K, I have 16,000 participants, and then I want to save them as CSV. So by saving it to CSV, it will save into my JupyterLab temporary storage, and then by using DX upload, it will be uploaded into my project storage into data folder. So if I look at the data folder, you see that the results are already here. Okay, so this was really quick demo. And just what are the next steps after this session? So you can go to Quick Start if you're not yet signed up or dispensed your project. So you can look at our tutorials. You can go to our newsletter. You can look into our Jupyter Notebooks. So they're not only the one that I showed, there are more of that. And we're actually very pleased that we now started a community forum. So we encourage you, in order to benefit not only for yourself, but for other scientists, to post the questions and some comments to your best practices there. So just as a summary of what we learned today is that JupyterLab file system storage is separate from project storage, and JupyterLab storage will disappear once session ends. So in order to use in JupyterLab in your JupyterLab files, you need to transfer them. So either you are using DX download, or you're using the XFuse file system by adding Manatee project, or and after you work with your files and you want to upload them to project, you are using the DX upload. Also, only one person can collaborate one person can work on the notebook. So other person will see it ad lock. And if you want to unlock it, you need to use kernel shutdown. And you can install some software packages, and you can then save them as a JupyterLab snapshot, and then can you reuse them in order to save your time. So I know that it was a dense session, but let's summarize the objectives that we learned. So we explained the relationship between JupyterLab and UKB-RAP project regarding storage. We select the appropriate JupyterLab instance given your workflow requirements. We also know how to access files in Jupyter Notebooks from project into JupyterLab environment. We can now run a Jupyter Notebook in JupyterLab environment and upload results back to project. And we know that if we want to install some software dependencies, we can then utilize them using snapshots. So I know there was a lot of information, but I really thank you for your attention. And I think that my colleagues already were answering on the questions.
Great. Thank you so much, Anastasia. Thank you for being our Sherlock, and Ted, thank you for being our Watson for this webinar. So I think, yes, thank you, Ted, for responding to the questions accordingly. And then I just wanted to point out, one of the best ways that we can get your help is if you can give us your feedback regarding the webinar, but also some future webinars that you would like for us to develop. So if you could please just take a minute or two. I popped the survey link in the chat. And as Anastasia pointed out, please feel free to use our community forum. That's community.dnanexus.com. Actually, I will go ahead and pop up in the chat the ways and the other resources that you can get help. So that includes the documentation, but also future webinars, and then a link to our community forum. I think I even popped, sorry, that was left for one of our webinars. If you have questions about the UK Biobank data, there's a link there as well. And so with that, I think, are there any questions?
So there's one open question. Well, there are a couple of open questions, but most of them are comments. But this one is from Santiago, and it says, I understand that you have to pay for storing your results in RAP. Can you download them from your project and RAP to your personal PC? Do you have to pay to download them?
Yeah, so you want to answer that on that, or?
Yeah, so I believe that you are able to download, like if you've generated like a CSV or TSV file, for example, you're able to download that directly onto your PC. You are charged for kind of extra files that you generate, although, depending on their size, it might not be that much. So you're not, as far as I know, you're not charged for downloading them.
Yeah, I just note that if you want to download, for example, whole exome data to your computer, that will not be possible.
Yeah.
Okay, and maybe we have one question here that maybe I would like to answer. That there was a person who was asking that for people who works with AI for creating phenotypes, selecting fields is not an option. And this is a perfect example when you need to go to a TED session to work with a Spark JupyterLab, when you don't need to select field by field, but there will be showing how you can select multiple fields at once. And by then you can actually then work and use AI for your phenotype creation.